Ladies and gentlemen, please welcome back to the stage, Audra Moran.
I just want to do that over and over again. Woo! And yell my name. I'm going to move this a little bit. Okay. Welcome back, everybody. I hope you had a great lunch and a great beginning to the day with your breakout sessions. We have more later on, and a lot of good ones. But right now, we're going to kick off our afternoon session, which is a conversation on clinical trials with Dr. Elise Kohn, who's here with us today, and who told me that she actually was at the very first OCNA conference 21 years ago, which is pretty amazing. How are you? Do you really not want an introduction?
I really don't want an introduction.
I have a lot of introduction that says she's awesome and really, really smart, and she doesn't want me to read it. So she's awesome and really, really smart, and I'm going to turn it over to her.
No. What I am is really grateful to be here because OCNA, OCRFA is special to me, and there are many seats where there's still someone there, sitting there who is in my heart, who I've known through all my many years. But one really awesome thing is that that first meeting was so small, and now there's so many of us, those of us who are working and worried about making progress, and you who are advocates and patients and family members. And normally I would go through the how many are, and so those of you who knew Allison, Allison was my cousin. And so if you knew Allison, who worked with SHARE, and she worked with survivors teaching students, and she continues to be part of that drive in my heart. But also all of you are, and the real question is, what can I do and what can we in the academic community do to make a difference? And that's really what today is about. So I was given this really cool title, but it's really two different topics. And so we're going to start out with a fairly simple discussion of clinical trials. And then I'm going to talk about three directions that I think are pretty exciting. And I will give you full disclosure now that after I wrote down those three ideas, when I was trying to put forward what the examples were, it happened to be that the National Cancer Institute intramural campus program was really leading in all three of those, especially one of the folks who I trained, who's now there, Dr. Chung-Min Lee. So as a proud mother, or maybe she would be my grandchild, I don't know, but I'm gonna brag. But her work and our work intramurally at NCI and the work that we at NCI fund is all moving rapidly, and it's extremely exciting. And I come from the NRG meeting, and I'm going to give you some hot off the press information. So as you know, I do work for the federal government, and the only disclosure I have is that I am not underworked and overpaid, as they think a few blocks away from here, but I don't have any disclosures. If we think about ovarian cancer in the 2.5 decades I've been involved in the field, and the many decades where our NCI has had a strong focus in ovarian cancer, we've seen the coming of platinums, which were the first real impactful intervention, impactful in how much benefit we can give patients, and also impactful in how much toxicity we can create. The combinations, the introduction of Taxol. The 2000s I'm calling the era of the angiogenesis inhibitors, and I like to think of this current decade as returning to our roots in DNA. So DNA is that stuff we have in every one of our cells that helps determine what color our hair is, and especially in my case, how early it started. And it's what we have in every cell, and it helps make us what we are, but then sometimes it goes awry and helps cause cancer. And drugs like platinum and Taxol, they're all things that cause damage to DNA, and that's why the tumor dies. And the new drugs, like the PARP inhibitors, block the process of fixing the DNA. So one is putting the pothole, and the other is digging the pothole deeper. So I think that this era could be considered the era of DNA repair, but I would be remiss, well, that's interesting. Here I am with my fancy stuff. There we go. And maybe it will be the era of immunotherapy. The new drugs that you hear about that have made a big hoo-ha in melanoma, lung cancer, kidney cancer, have not to date been overwhelming in ovarian, in gynecologic cancer, other than the subset of endometrial cancers. There may be some interesting information coming out of NRG in the next couple months about one trial that's just recently matured. It'll probably be reported at the International Gynecologic Cancer Society in Kyoto in the fall. But mostly, we're still trying to figure out immunotherapy. So let's step back and think about all this. And this is just going to be 101, which means many of you are going to get bored. If you are, as long as you don't snore louder than I talk, we're fine. And before I go any further, so here are the ground rules. You can interrupt me at any time as long as I can see you with the spotlights, but somebody has to keep track of where I am because I'm only a few months out of chemo myself, and I get chemo brain. So if you do interrupt me with a question, a few of you who know me, okay, so WHO, the World Health Organization, says a clinical trial is a research study that plans ahead and assigns participants to one or more health-related interventions to evaluate the effects on health outcomes, which is a whole lot of syllables to say. We give you a chance to have something that you wouldn't get ordinarily, and we do it in a controlled fashion where we collect information from safety to outcome so we can decide if it's worthwhile moving forward or not. More words, fewer syllables. Why clinical trials? Well, it allows us to plan ahead on how we want to look at something so that we have careful metrics, and so we can interpret what we find, and we can apply that to drugs, to radiation, to imaging, to surgery. Oh my God, we had a proposal for HIPEC last at NRG. Complementary and alternative medicines and modalities should properly be tested in a clinical trial because you don't know when you're doing harm. Remember, it's not just about benefit. Sometimes you learn about harm, and that's an important element if you're a patient. How many have neuropathy like me? All right, yeah. So having harm is an important thing for my other side of the equation to be attentive to. The clinical trial phases that we're going to talk about were developed so that we have a common language across investigators and even across countries in a way that we can formalize the objectives and the steps we take so we're not jumping too far, so that we can minimize the number of stoic leaders, volunteers who want to participate. Because when you participate in a trial, there's no guarantee, right? You're really being a pioneer who's hoping for benefit, but you're still being a pioneer, and there's a due diligence on our part. So we want to minimize the number of patients, which is sometimes annoying as patients, but we want to be sure that we're balancing that safety. And we want to provide evidence so that any physician in an ivory tower, or as my old mentor, Eddie Reed, some of you knew Eddie, used to say, if you're a private slick on Rockville Pike, and Rockville Pike is that big road that goes through suburban Montgomery County with all the strip malls on it. And so he was just saying, it doesn't matter if you're a private practitioner or you're the head of one of the biggest research institutes. The point is that you're still going to be making individual decisions, and you want to do that with evidence. Anybody can perform a trial. Ideally, they need to follow those guidelines. They must follow the regulatory guidelines, which are also in place for safety. NIH, my umbrella, we fund grants and contracts in many disciplines of medicine. Obviously, we fund a lot in oncology through the National Cancer Institute. We also have the intramural program on campus, which is a separate program, and some of you have heard about it. Some of you may have been on our trials. Those are done at our clinical center in Bethesda, a few metro stops up the road. All of different components of pharmacy, clinical research organizations, academic institutions, lots of different people can run clinical trials. Force has run trials of information gathering. Remember Cindy Melanson? Some of you? Yeah. Cindy did a questionnaire. It was a trial of sorts. It was one of the rare times where 500 went out and 1,500 came back. I tell that to the academics. Okay. So who participates in clinical trials? anybody who's interested, willing, and fulfills the eligibility criteria, which means that everyone should. Now, that's ideal from my point of view. In children's oncology, over 90% of children are on clinical trials because we fight for our kids, right? And we want to take them to the ivory tower and make sure. In adult oncology, that number is terribly low. And that's because many of us have multiple jobs. We're mothers, we're daughters, we're parents, we're teachers, we're professionals. It doesn't matter what we're doing, but sometimes we don't have that breadth of time. But I still want to encourage everyone to think about trials. The clinical trial phases test different questions from phase 1 to phase 3. And I'm not going to go beyond that because those are post-regulatory. We look at the objectives. We make sure the eligibility fits the question we're asking as broadly as we can. We design the trial approach, size, and endpoints to be as accurate and as precise as we can to minimize risk and maximize benefit and information, and to guide the next steps, because ultimately the goal of a clinical trial is to bring something to everybody, to bring a drug or an idea to everyone. So phase 1 trials are really about safety. Sometimes they're open to a wide variety of cancer types. Sometimes they're very selective or specific. Depends on what question is being asked. Sometimes we're adding drugs together because it's uniquely important, we think, in ovarian cancer. Sometimes it's a first in human and first come, first serve. Phase 2, we always want a more homogeneous focus of patients because we want to be able to move towards that next step of targeting regulatory approval. We have a predefined dose and schedule from phase 1 most often that we're ready to use and we feel safety with. We're looking for signs of activity as a primary endpoint, and sometimes they're randomized. Oftentimes it's between different kinds of regimens or controls, rarely but occasionally with a placebo in a phase 2. So that's a question to ask if it's a randomized phase 2 that you're looking at. Is this a placebo or are there 2 treatment arms that are asking different important questions? And in phase 3, that's often when we're specifically looking at the new intervention and targeting it for regulatory approval by the FDA, which means that we're comparing it to an accepted standard. In many cases, that standard may not always be what you and I perceive as the immediate standard. For example, bevacizumab is now approved, I think, from birth to death in ovarian cancer. You know, it's sort of in a wide paintbrush, which means that there are going to be some trials that may not have bevacizumab in them, but that's not necessarily bad because there are many other opportunities to get bevacizumab if it's not in that particular part of your treatment regimen. The intent of a phase 3 trial is to change practice. So it's part of that informing the doctors and patients for the future. And we can do it by showing something's better, which is the fewest number of patients, or occasionally we say we want to make sure it's not worse than, which is a problem because we get into too many negatives in the sentences and it gets confusing. But sometimes we're trying to show something is not worse than the standard of care. Sometimes they're blinded, sometimes they're not blinded, sometimes there's placebos, and sometimes they're not. It's hard to blind something when one arm is oral and one arm is IV. How many of us would volunteer to get poked if we didn't need to? It doesn't matter that I still have my port. You know, I go around like this every day to protect it, but it doesn't mean I want someone to poke it if I don't need it. So if it's going to unblind itself, there's no reason to subject any of us to that additional. So I'm bad. Sometimes I have to make myself laugh, so I have a few cartoons. After reading about all about cancer, treatments, trials, phases, and stages, I've decided to quit reading. There are days I feel like that, and for those who can't see it, the cat says me too, and the thing he's reading is the NY Trials, New York. I thought that was cute. Okay, so what goes into designing a trial? I think my institute thinks, and I think we all should demand scientific justification. Not just because a drug is there, we should test it, which unfortunately is how a lot of things are happening. Are there data? So my cartoon We found a mass. The good news is we have weapons of mass destruction. And we do in ovarian cancer have weapons of mass destruction. Isn't that awesome? We don't need anybody to come check it out. We know we've got it. We have to have the potential to make a clinically meaningful finding. Once upon a time, it was clinically meaningful to test carbo versus cis. We know they have different patterns of toxicity. Everybody says carboplatin's not toxic. We know that's not true, right? It's just silent. It knocks out your bone marrow slowly, right? We all know that. Well, tell some of my counter. So, but nowadays, if someone came into it to me with that trial, it would be Tweedledee and Tweedledum. I would say there's got to be a better question to ask because we have a responsibility to you, our constituents. I think of you as not as patients, but as friends and constituents. My job is to make a difference with you and for you. If I think of you as a patient, I'm not necessarily with you. It has to be interesting to you, though, because you're going to vote with your feet, which is my cartoon. You're going to vote with your feet. If it's not interesting or you perceive it as not safe or not pertinent, you're not going to participate, and it's going to languish, right? Of course. And lastly, it has to be feasible. I can't swallow that. How many remember olaparib capsules? Oh, my goodness, yes. The handful that you had to take. They actually had less toxicity than the tablets. The nausea and stuff is actually a little higher with tablets, but it was daunting because not only were there 8 of them, but they were big. So whatever we do has to make sense and has to be feasible. And as someone who just came off of a large blue one, I understand that. And so those are the criteria that I think are important in going into clinical trials. And so that was my lightning run, Clinical Trials 101. Going to move on to some exciting new directions. I thought there were 3 that were important. I thought I wanted to share. I think that we as a scientific community have to start understanding PARP inhibitor resistance and how to use PARP inhibitors after someone has already had a PARP inhibitor. But that is short-sighted in that it forgets one segment of our community. Women who are not carrying a deleterious germline mutation, women who don't have an HRD positive tumor, and especially women who have disease that is now platinum resistant. How can we make PARP inhibitors active for everyone? I'm going to talk about the olaparib cediranib story and give you some hot news. Leveraging immunotherapy, I think that we all on my side of the aisle realize it's time to do combinations because immunotherapy a la melanoma and lung cancer doesn't work by itself. I apologize, I'm really dry. And the other is the new direction of modifying the cell cycle. Okay, breast cancer got there first with the CDK4/6 inhibitors, but we got a hot one for you with the CHEK inhibitor. And as I said, as a proud mom, all 3 of these emanate in part from the intramural NCI and my team and those who my juniors who are now in charge. So many years ago, we were playing with olaparib in the lab, and my colleague wrote a paper that suggested that DNA repair, the target of PARP inhibitors, was necessary for angiogenesis, for blood vessel development. And they did it in an eye model that was the model that helped us understand how premature babies lose their sight. That same model. And wow, I got to put an angiogenesis inhibitor and olaparib together. So actually, we did, and it worked. Nonetheless, we are now in a position where we're testing it. And it turns out the way it might work, and don't worry about the graph, I had to put a picture in there because I hate talky slides, was that if you take oxygen away from the tumor, hypoxia is our term for that. You know, we always have to have more syllables. So if you take the oxygen away from the tumor, it actually turns off BRCA1 and BRCA2 and some of those important components of repairing DNA. So making the tumor oxygen hungry seems to make it sensitive to PARP inhibitors. And the picture, look at it as modern art. In the upper left, if you don't treat any drugs, you get that scribble of lines. Those are actually blood vessel cells coalescing in the laboratory model to make blood vessels. In the bottom right, If you add both a PARP inhibitor and an angiogenesis inhibitor in doses way lower than we can get when we take these drugs, you don't see any squiggly lines. And that was what we did in my lab. And that has led ultimately to the clinical trial you heard about a couple years ago of olaparib in blue versus olaparib with cediranib in red. And what we found is it was dramatically better overall, twice as much. What was especially exciting is on the right, which is women who aren't BRCA mutation carriers did better. And that has led us to 2 clinical trials. NRG GY004 for women with platinum-sensitive disease. This is fully accrued and it should report out first or second quarter 2019, we hope. And we're hoping that it will show the combination is better than platinum-based chemotherapy. And also, we have tested it in women with platinum-resistant disease because that is a group of women where we have fewer women with germline BRCA mutations. And if you look on the right side, this is fresh data, look at the number of lines below the horizontal, and what we're seeing are more benefit in this group of women than we would expect. The response rate of single-agent PARP inhibitors in women with platinum-resistant disease is in the single digits. And at an early point when they did this analysis for ASCO, it was already at 20% in this little trial and rising. And we have just found out that GY005, which is testing single olaparib, single cediranib, the doublet, and platinum-based therapy, we did a pilot, the phase 2, and we had to wait a year for it to mature. And we just found out that on Thursday morning, we have the go-ahead to reopen the phase 3, which will just be platinum-based therapy, which is the control, cediranib by itself, and the combination. We hope that'll be open by October. There's just some regulatory things that we have to do to get it reopened. And the goal for both of these studies is to bring this combination for regulatory approval so that you can get it routinely. We also have an app we created. It was just published recently in one of the JCO journals that goes on a smartphone that helps monitor blood pressure and diarrhea, which are the 2 major toxicities. We're going to be piloting that in GY005, initially at 16 sites, and then hoping to broaden it, because that's a way where you as the consumer can communicate that toxicity real-time, get real-time feedback and real-time direction. And we think that's an important, that kind of interaction, that kind of electronic communication is an advance that we hope is going to be taken up more broadly across oncology. Immunotherapy. So my thought is, if you can get 1 + 1 = 3, you might flunk algebra, but as an oncologist, that makes me happy. So one of the things that we know is that if your DNA is getting bunged up, whether it's by having a mismatch repair defect like women and men with Lynch syndrome have, or germline BRCA, or a damaging drug, that's going to make more non-self proteins in your body, and then your immune system's going to be tickled. And it's felt that those not me proteins in your body are what are part of activating and reactivating the immune system. And when you give these new immunotherapy drugs, what you're trying to do is goose the immune system to recognize them. So there's 2 sides to the equation. So we thought that if you used a drug to wake up the immune system, those immunotherapy drugs you hear about, and you do something to create more stimuli internally by using a PARP inhibitor or an angiogenesis inhibitor, then 1 + 1 may be more than 2. So Dr. Lee, my colleague, was the first to do that, and we did phase 1 with durvalumab, which is an anti-PD-L1. It has a name now, Infinzi, I think. We're not allowed to use trade names, so I never remember them, and they're really complicated. But durvalumab and olaparib, or durvalumab and the angiogenesis inhibitor. Everything below that little red line and long out the horizontal line is good. And what you see is a moderate amount of good, even in the phase 1 study. We had an early signal that this is a good direction to go. And we're pretty excited that we've seen some women who have a couple who have had their disease disappear. And we also think it might work better where there's already an immune response, but what we're trying to do in oncology in general, the new term is to make a cold tumor hot, a tumor that doesn't have a lot of immune, and you may have heard it this morning, I guess, from people who are, tumors that don't have a lot of immune cells in them, which are shown in brown here, that you want to maybe pull those in. And one way to pull those in is to goose them and give them a reason to be there. So the drugs that we're trying to use. We also are trying the triplet, because if 2 injury agents are good, maybe that, and we see activity with olaparib, cediranib, maybe that's even better. So in our tiny pilot, it's really interesting, and it is working in women also with platinum-resistant disease, which is an area that we have an unmet need. We're really, in my mind, is a very important area. So you can see some progress. And after we started, the group up in Philly also put durvalumab and olaparib together just for women who had germline BRCA mutated association disease. Mediola was presented at SGO. Again, the longer the line, the more interesting it is, showing promising results. The longer it goes down, the better off it is. Again, promising results. And the last bit before I take another sip of water and let you have at me is to tell you about the cell cycle modifiers. So for cells to grow and divide to make tumors bigger, they have to go through a cell cycle. They have to divide and make 2 daughter cells. And in the process of going through that, we normally have mutations which we fix. That's just part of life. We mutate normally. It's hard to tell, or maybe I am a mutation, but you collect mutations. And normally what happens is the cell stops when it detects too many and fixes them up and then keeps going again. But if you pushed that cell through that door and said, no time to repair, keep going, then it's going to accumulate those injuries and ultimately die. And that's what some of these new inhibitors do. If cells can't stop their growth cycle, they accumulate injury and they die. Dr. Lee had this really great idea to mix a check 1 for cell cycle checkpoint. There's check 1 and check 2. She wanted to mix it with another drug and had a lot of support for it, and then the drug fell out. And she said, what am I going to do? And I said, go find another one. And she did. And Prexasertib, or LY386, I'm always glad when they name them because it's much easier to do informed consent when I have a name. We call it Prexa for short. Prexa is a very early development agent that we were very pleased that Lilly allowed her to examine in an investigator-initiated trial. We thought it might be better for women with BRCA germline-associated disease. So we had 1 cohort of women who had germline BRCA mutation and 1 who didn't. We finished the cohort of those women who didn't first. We didn't care if it was platinum-sensitive or platinum-resistant. We're into all comers. You've had 6 priors. If you're eligible, you're eligible. We're pretty open-minded, and despite the fact that we're always accused of being selective. And in this case, on this graph, going down and going out is good. And what do you notice? Most of them are going down, and most of them are going out. And this is early data. This was out to 17 months. Most of these women, the lines are gray, which are women with platinum-resistant disease. This was published in February in Lancet Oncology. It's a really exciting drug. Lilly has just completed, I think they accrued in record time, a multi-institution, multi-country study to confirm our findings, and this will hopefully be going into phase 3 soon. I really think, and this is my opinion, not the opinion of the National Cancer Institute, I think this is a hot drug, and I'm excited about it. We're examining it in other cohorts, and Dr. Lee also has a combination trial, so we have opportunities on all of these trials, either presently or coming soon. So, in the old days, we looked at chemotherapy, we looked at radiation, things that were DNA-damaging agents, and we had benefit. but we really didn't have cure. And we thought we had improvement, and we were lengthening lives, but now when we add these DNA repair inhibitors that also cause harm, we're definitely seeing periods of benefit. We're seeing women whose disease isn't progressing for longer. And while some of that hasn't translated into overall survival, that's hard to interpret because we know we're all living longer for a variety of reasons. Maybe if we can now affect the local environment either with stimulating the immune system or reducing local oxygenation to increase injury, maybe we're where we need to be. And so with that, the mouse says, I go home today, they cured me using this new miracle drug. I'm afraid it's years before it's approved for humans. And the good news is even that many drugs are getting to patients sooner and faster and more and carefully. And there's a lot of options out there. So it's important to be sure to ask good questions. Make sure it's right for you or your loved one. And hopefully this was wasn't too rapid, but this gives us plenty of time for broad discussion and afterwards. So does that sound good? Ready to?
Thank you. Okay, we do have some questions. So we have a few questions that center around the same topic. Last year at this meeting, Dr. Carol Brown spoke about clinical trials and talked some about what seems to be a drop-off in the number of clinical trials. And so some people are looking for an update on what's going on with clinical trials in ovarian cancer and gynecologic cancer.
So first of all, there has been some reduction in total number of trials. And part of that is because there used to be a lot of big trials, all-comer trials that were very large, where it didn't matter if you had a germline BRCA mutation, whether you had high-grade serous or low-grade serous or clear cell cancer. We just put everything together. And then we tested 3 cycles or 6 cycles, or IP versus IV, and we did those sorts of things. All those were sort of coming to closure around the same time, which left a vacuum. And it happened to be at the same time that the way in which the groups were organized and funded was changing in response to the big IOM report. And so you got to blame somebody. Why not blame the government? And then the other side of that coin is that there's been this statement that we went from 7,000 patients to, well, the 7,000 whatever number of patients on trial included patients who are participating in chemoprevention trials, patients who are only providing tissue for banked trials. But if you look at the actual number of women who are participating with gynecologic cancer who are participating in treatment trials, that number actually didn't drop as dramatically as has been argued. Now, that said, since the new organization in 2014, we're now up to 20 new gynecologic cancer trials, 3 or 4 of which are opening in the next few weeks or so. There's a new trial in clear cell ovarian cancer that's small but opening. There's a trial in cervical cancer that's coming down the pike shortly. Another one, actually, 2 in clear cell cancer. One is a new drug called tazemetostat. We have a randomized phase 2 in endometrial cancer that's going to be looking at a variety of arms that we're going to be able to loop new arms into. So that's another thing. If you create a mechanism where you can bring new arms into it, it doesn't count as a new trial, does it? Depends on how you count. So I think the answer is that, yes, there was a slowdown. It was a natural event because of what was going on at the time. There are fewer large monster phase 3s because at least in the NCI-sponsored trials and even a number of the company-sponsored trials in industry, we're looking to make the treatment fit our population more appropriately. And those are going to be more randomized, more often I think we're going to be randomized phase 2s. They are not going to be 700 patients, which means that they'll get done faster, we hope. And because everybody is doing better, living longer, we are more often using progression-free survival endpoints, which means that we don't always know if in the long run the benefit makes a difference in overall survival. So that means we have to be cautious about how we as physicians present things. So on one of our trials, when all the PARP inhibitors came out and available for women who are second line or further for maintenance, patients who were on the olaparib cediranib trial for platinum-sensitive patients, there was all of a sudden getting olaparib, which was a protocol violation. Well, olaparib may cause a progression-free survival benefit, but we don't know that there's an overall survival benefit. And the original study, Study 19, didn't show that. It's the only one truly mature. And so it can still... There's still access to those drugs for treatment and later maintenance. And so it's important that we on the clinical trial side explain what benefits and risks. You know, bevacizumab is really now available frontline, platinum sensitive, platinum resistant. You ask me when I would give it in platinum resistant because I'm getting more bang for the buck for you when I do it then. And the biggest bang for the buck is when you've never seen it before. So part of it is parsing what's now this tremendous collection of opportunities to figure out how to use the drugs best when. And that we talked about at the NRG meeting is also critical in deciding how to design the trials and also how we communicate with and educate patients who are interested. And so what we're going to see maybe are fewer but smarter and more interesting trials. Unfortunately, that's not completely true because there are 5 trials starting in Europe, overlapping without prior data per se, testing IO combinations, the immunotherapy in front line. And that's also because in industry, there's a fight to the finish. 5,000 patients, 4 trials, all pretty much identical, different drugs. Annie. Yeah. So it's frustrating. So there's a difference between how industry looks at trials as well as academia. And the NCI's position is we want to do it smart. We want to have a scientific reason. We want you to be participating in something that makes good sense and has a chance to benefit you as much as possible for the type of ovarian cancer or any cancer that you have, because one size doesn't fit all anymore. And so that also addresses the issue, and I covered a couple other topics, and I'm sorry, but I think it's a big issue, and I wanted to be sure that you realize that this is not something to be angry about, but to realize that this is something that we're heading in the right direction, in the smart direction.
In what circumstances would you get a placebo in a clinical trial?
Great question, and I should have covered that. Thank you. A placebo allows the doctors and the patients to not know which arm, and that means that the doctor is maybe not going to look extra carefully or differently at women or a patient on one arm versus another. Also, in the setting, for example, of the olaparib and cediranib, we couldn't placebo control it because the chemo was IV for however many cycles were necessary, and olaparib, cediranib, and olaparib alone are all oral. Can't control that really. And also the toxicities will unmask. But that whole thing about giving that olaparib, which was before the primary endpoint but off trial, and if there's a placebo, you don't know if you're getting that agent or not. So everybody has an equal shot at it. The doctors are looking at each individual patient with the same level of caution and following the protocol. As a patient, it can be uncomfortable. I understand that. That is an issue I can't get around, but what I can say is it's important as a patient to understand if you participate and you are on the placebo or not, what are your benefits and what are your risks? Are you missing something that you might never be able to get again? In the very first Taxol trial in ovarian cancer, you couldn't get Taxol later. It was the only clean trial. In Europe and in Canada, if it isn't licensed for a particular indication, you cannot get it. In the United States, all you have to do is burp and you can get it. So that means that it's very hard to run a clinical trial that's clean, making placebo even more important. To get data that provides the evidence, that means that we can tell you with the greatest confidence honesty and evidence whether or not something we're doing is going to be safe and effective for you.
What's the best way to find a clinical trial if my doctor does not seem particularly well informed?
So, clinical trials. Oh, I love this. This is my bully pulpit. Annie's laughing. Clinical trials are not, and excuse this expression, but you'll understand why, are not for women or men who are damn near dead. Clinical trials are always the front line. So even for newly diagnosed patients, it's always worth asking, even though it's a traumatic time, and it's always traumatic, but it's more traumatic at the first run. So there are many ways for you as a consumer, as a patient, as a family member, to go after it. And you should and could be your own advocate or your family's advocate. So, of course, there's 1-800-4-CANCER. The OCRFA has a clinical trials matching service at their website, www.ocrfa.org. The NCI is 1-800-4-CANCER is the phone number, or clinicaltrials.gov. If you're interested in our intramural program, like adult, and I can't even blame this on ChemoBrain, I didn't put in my slide that I usually do to proselytize our trials up the road. But you're welcome to email me. Or you can get us off of the 1-800-4-CANCER or clinicaltrials.gov. Our trials show up. Every trial that has federal funding has to be listed in clinicaltrials.gov, and you will also find them off of the OCRFA website as well. You will actually find a lot of industry trials too. It's probably the most comprehensive resource. If you call the 800 number, you get a human being. If you try me, I will do my best to get back to you, but I will warn you, the next few days are going to be rough for me, so it may not be as timely as you would like. And I'm going to try, I've got all but the last digit, so I will look it up and make sure that I get it to Sarah for connections for the link to the intramural program.
What are some questions I should ask my doctor when deciding about going on a clinical trial?
Why should I have what you can give me? Instead of saying, why should I go on a clinical trial? You can get insurance to pay for that anytime, right? Why should I miss an opportunity if there's a clinical trial taking the standard of care when the standard of care is always going to be available, certainly pending my health status? Turn the question around so that you're advocating for yourself. That would be one. Is there a trial, any trial close by? Because there's a lot of toxicity, not just neuropathy and blood counts, right? Time away from work, transportation. I have to have someone drive me every time I go in, so I understand what that's like. So some places, everybody thinks Mayo and MD Anderson and whatever, that, the Holy Grails, but the Holy Grails are sometimes pretty expensive to get to. Some of them, like Memorial, don't take insurance. Those are all fair questions. At the NCI, if you're on trial, everything is paid by your tax dollars. If you come up to our center up in Bethesda, I think it's fair to ask what the expectations are of that trial. If the doctor knows where the toxicity is, or if your doctor knows how you might reach out to get more information if he or she doesn't know. Also, go back to the OCRFA website and links, call 1-800-4-CANCER. If your doctor doesn't know, sometimes I've had patients who go into their doctor, already printed things off of the web, said, look at all of these, are any of these okay for me? Because, oftentimes your doctor is seeing you, 10 minutes? If you're lucky, 15, it's very fast. And if they're seeing a lot of patients, they don't always have the time to do that footwork, which is unacceptable at one level and unavoidable at another. So don't be shy about doing the footwork and bringing it in as well. Oftentimes I've found patients who have said, I took this to my doctor, and they said, oh, that's a great idea. And then they helped out. So when we as patients are advocates for ourselves and also our friends and family, we make the biggest step forward.
I just want to circle back briefly to this idea of placebos and trials, just to make sure everybody is very clear. I think sometimes people are concerned that they're going to go on a trial and they might get a placebo when in fact they need treatment. And so they want to make sure that going on a trial, if there's a placebo involved, they want to make sure that it doesn't mean that they're not going to get treatment that they need.
That's a good question, Sarah.
So just, can you speak to how in what circumstances would a trial even have a placebo component?
So any trial that's randomized isn't going to be approved through regulatory and IRBs, whose job is patient safety, if there's not some perceived balance between the arms. So even if there's a placebo, there is a sense that at a minimum, you're getting reasonable and appropriate care for where you are in your disease setting and your overall health. And that whatever the placebo is blinding, the hypothesis is that that may further enhance your outcome, although it may also provide some toxicity. The goal here being that you're getting at least appropriate standard of care. There are settings where that could be observation, in which case it's just the placebo, although I'm not sure we have any going on in ovarian cancer at all like that right now. But at minimum, you're getting standard of care. The hope is you're getting better if the experimental agent does what's expected. At every level, there are checks and balances, whether it's the regulatory FDA level or the IRB where it's patient safety. There's all those elements to make sure that there's that reasonable balance. And if you're not comfortable and you're not sure, that's exactly the question to ask. What am I going to lose if I participate and I'm on the placebo arm? Will you unblind me so I can get this agent if it turns out to be useful? And that's also an important question.
What does it mean when a clinical trial has been discontinued due to AE?
A&E is not arts and entertainment, and it's not good for movies. It's adverse and event. So an adverse event can be anything from, I don't feel quite perfect today, grade 1 fatigue, to my blood counts are 0, I've got a fever of 103, and I'm in the intensive care unit, which is a grade 4. Adverse events are what we in clinical trials we take events that occur, anything that you tell us about or we observe in labs or your clinical, and there's a set mechanism for determining level of severity. And there are criteria that are used across the US and across the world, and the NCI is pretty much the one who sets those guidance. And those criteria across the board are used that a certain degree of adverse events or serious adverse events, toxicities that cause patients to become ill or to discontinue or reduce therapy, there's a certain allowable amount, and there's a point at which that is too great a risk. Now, I've had someone come up to me before and say, why can't I get on this phase 1 study? I have cancer. I'm doing poorly. What does it matter if I have these other things? And well, if someone is ill and has more compromise of different organs, she's more likely to have toxicity and more likely to be harmed by the experimental drug. And our tenet is first do no harm. So that's why we have eligibility. But once you're on a trial, we have to monitor, because if we start to see, like with the immune therapies, people lose their thyroid function. Well, a lot of women would say, so what? You know, how many are on thyroid replacement by the time they're... But it also can cause lung changes that are progressive and can be harmful and even life-threatening. It can cause bloody diarrhea, colitis, which can be life-threatening. So adverse events, if they reach a threshold of severity and or frequency, can lead to discontinuation because the safety bar has been reached, whereby we feel the possibility of benefit is overcome by the data indicating harm.
Okay, so we have some questions about cediranib
Yep,
side effects.
yep.
What are they? One person wants to know. And what can be done to manage them? They've been described as horrific and life-altering. Oh, card. Is anything being done to try to minimize those side effects?
Horrific and life-altering. I think having cancer is horrific and life-altering. Right? But we're all here to say that we can survive that, right? All right. Cediranib is a small molecule that inhibits the part of the cell that recognizes the protein called VEGF, which is what Avastin sucks up when it's floating around. And so it's one of the angiogenesis inhibitors. It's in the same category, if you follow cancer broadly, as sorafenib and lenvatinib and sunitinib. They all have the similarity of inhibiting the VEGF receptors. Cediranib, it's kind of blunt. It's in your face. The side effects start within 24 to 48 hours. It doesn't hide. It doesn't come up on you gradually, which is probably good, because we tell patients, within 24 to 48 hours, your blood pressure is going to go up, and we're not kidding. We're giving you the prescription. We're giving you the instructions to fill it before you start. We're giving you a blood pressure cuff, and we're giving you parameters. If you follow this, that'll be fine. And in fact, with the app, as we were piloting it, it's a blood pressure cuff that's Bluetooth to the app on the phone. And so from the investigator side, it goes right into the source document, but the patient sees her blood pressure, and then there are flags about what to do. And that's why we did that app, because we know that the blood pressure is a real problem, and it's fast, and it's early. And the good news is if you stop cediranib, it goes back down. But we don't want you to do that because we think it's a good drug. It also causes diarrhea, pretty much the same way. In our adverse event monitoring, a change of 4 or more poos per day is grade 2, and we worry about that. But we tell you, when you get cediranib, we say, listen, you're going to get diarrhea. Now, how many patients have constipation who come to see me? It's amazing. So, in part, we're helping, right? If we can't laugh, it's not worth getting up. But we give a prescription for Lamodal. We give you a plug, and we give you instructions on how to take it. And that helps. Unfortunately, there are rare events if someone doesn't take their plug, they don't take their the Lamodal the way we instruct, then it can be a problem, and there's diarrhea and dehydration, and you feel lousy, and you end up in the ER. Even if you do it right, sometimes that happens, but not so commonly. The app is great because you put in how many poos, and it says, brat diet, call your doctor, and it shows a picture of bananas, rice, toast, and applesauce. And I was thinking, yeah, but most of us are mothers and or aunties or grandmothers, and we all pretty much know what the brat diet is. But they showed it anyhow. And it's on the phone, on the app, which is kind of cute, but it reinforces it. So these are real side effects, but they can be gotten around. And when Dr. Lee was examining cediranib with the immune therapy drug durvalumab, it was a big concern for us because all of the immunotherapy things that are hot and in the news, they all have a risk of diarrhea and colitis, where you get inflammation of the colon that can be dangerous. So here we had 2 drugs that can cause diarrhea, so we had to be very sensitive to it. And it turns out we went to 5 days on and 2 days off with cediranib. It was like another world. Much, much better, really better tolerated. And I'm going to advocate that that's the dose that we use in the phase 3. I'll see if I can get that past the FDA. But it's a good drug and it's a good combination. And as we all know, we recognize when we're on treatment that we're giving up some control, some freedom, some of our quality of life balance and we choose that as individuals, how much that. And after a while, when you're on something oral, instead of the highs and lows of chemo, where you feel really yucky, and then so-so, and then you feel great, and then you get hit again, when you're on an oral, it's a more consistent process, having just come off a year of something like that. But mine was 3 weeks on and 1 week off, and so it was a little bit more like chemo, because that last week was the week. But I understand that as a patient as well. And so I think the 5 and 2 may help out, but we think it's still an interesting and effective agent, and I'm hoping that we'll find that out. So stay tuned. We're trying to reduce the adverse events and find ways to get around that, okay?
All right, we're running out of time, so maybe just 2 more questions. This one just came in and it's a good one. With so many combinations being studied, how does one maintain clinical trial eligibility for the future?
I thought you were going to say, how does one pick? I'm so glad you didn't ask that. If it's in there, tear it up. No. No.
That's my next question.
Don't. There's 2 things to say about that. One is that clinical trial eligibility, we try and keep broad, and then after a recent workshop between the NCI and ASCO and other stakeholders, there were further discussion on how to have more general, open, real-life eligibility to broaden eligibility as much as possible, which we're trying to do. The only time at NCI where we will put in selective eligibility barriers is when we get into safety. If a drug causes irritation of the liver, causes liver enzymes to rise, if someone who already has that goes on, how do I know if I'm making her liver worse or it's just a transient effect of the disease? So some of those we parse out, but we work very hard at the NCI to make sure that, especially since that recent set of guidelines, that we're trying to make sure that eligibility is broad so that women and men can maintain their eligibility across. Generally speaking, if you are active most of the day, if your labs are generally pretty good, you're able to eat and drink, do your activities of daily living, you're going to be an ECOG, at least an ECOG 2. All of you who are here are clearly much more, have a better functional level. And what we're trying to do is make sure that at least patients who fit that bill, if you're on oxygen, a lot of these drugs can cause lung injury. So I wouldn't want to put you on one of these drugs where I might harm you and hasten your pulmonary problems. I'd rather get another 100 or 400 patient experience, right? That's the whole thing about parsing those clinical trial steps, because if we don't know, then we can actually do quite severe harm. And one of the things that we always look at, there are always, unfortunately, men and women who die when they're on a study. Most often it's because of their disease, but not always. And so we, in terms of keeping eligibility, when the tumor takes over or side effects take over in a dangerous way, sometimes we can actually work around that with medication and other things. And sometimes we can't. And in those settings, we can do way much more harm. And that's always a tough question and a tough balance for us as physicians.
Okay, this will be our last one. What do you think of this whole right to try business for patients who have not had success with existing standard of care drugs? Does it mean anything for ovarian cancer patients? What's it all mean?
Okay, so this is my personal opinion. Although ASCO, most of the organizations have come out with the same opinion, I think it's dangerous, because what's happening is oftentimes neither the doctors nor the patients know the history of the drug, how it works, why it's toxic, why it doesn't work, and that can create a lot of harm. I think there are circumstances, especially where the physician and the patients do have the knowledge to understand, and there are circumstances that make complete sense. The rare case of ovarian cancer where we might find Lynch syndrome, mismatch repair, well, absolutely that patient should get an immune oncology agent on trial or not. But that's also going to be within the FDA guidance, the recent guidance, which was disease agnostic. So I understand it as a patient, because yeah, I'd be frustrated if there was something that could cure me and I couldn't get access to it. But the only thing that might cure what I have and I have lymphoma, are the CAR T cells, which is hype, 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 right? But I also know that the mortality for CAR T cells is over 10 to 20%. So I have the right to try that theoretically, but I'm not ready to give a 1 in 5 chance of dying. I got a 19-year-old who's a handful. I got to be around for a while. So I also have, I hope, more than 6 months, because I got a lot on my mission plate still. But I think we have to be really, really cautious, because there's a lot of hype out there. Just watch TV. Yervoy! You read in JCO. Well, it's 4.7 months versus 7.2 months on average. Well, is it worth going to all that to get your thyroid knocked out and have diarrhea and stuff for an average of 3 months? I don't know. Everybody has to figure out that. But the other thing is, most often when patients are truly ill enough with their disease that they really are facing those last months, which is part of the right to try law, 6 months, I believe. I haven't read all the details. Very, very few things work. That's a setting where we cause way more harm than good, whether it's standard of care or investigational. So it's a hard choice. I realize that. And as a patient, I appreciate things sometimes differently, because when I find my masses, and there's a few of you who've been through it a few times with me, and I fall apart like everybody else does. And then I read, and I try not to read about what I have, but when something new comes out, like after ASCO, I said, oh, I just had that. Oh, acute leukemia, 7% in the arm of what I just had and 10% in the arm of what I've already had. Does that mean I have a 17% chance of getting leukemia? Or a 7 times 10%, 70%? So I think we have to look at things as an individual. We have to balance risk. We have to not give unacceptable, unsupportable hope. Hope is critical all the time. But to mislead someone with hype and send them down a path that may cause harm because there's a belief that if you don't get it, you will die, I think is kind of risky. And I just worry that many people could be harmed from this law. I'm hoping more people will be benefited than not. But I'm worried, and I think that's the position across the board of pharma, the academic institutions, ASCO, SGO. I think most of us have all come out with a lot of the same positions, and as an individual and as a doctor and as a patient, all those things I'm kind of concerned.
We're out of time. Thank you very much, Dr. Kohn.
You've all been wonderful.